So next speaker will be Dr. Steve Henikoff. Dr. Henikoff is a professor in the Division of Basic Sciences at the Fred Hutch Cancer Center and the Howard-Hughes Medical Institute Investigator. He received his PhD from Harvard University working in the lab of Matthew Meselson. He's a member of the U.S. National Academy of Sciences at the American Academy of Arts and Sciences. And he was elected an AAAS Fellow in 2012. Dr. Henikoff and his lab developed key methods for sequence comparison and genome annotation, including BLOSUM substitution matrices for protein sequence alignment, produced in collaboration with Jorja Henikoff. His lab focuses on understanding epigenetic mechanisms involved in gene regulation and has developed widely used epigenetic profiling technologies, including CUT&RUN. So now let's please have a round of applause to welcome.
Okay, I'd first like to thank King and the other organizers for putting on this celebration and for inviting me to be part of it. I thought I'd tell you a little bit about when I first met Gary. It was actually at the Finding Genes workshop at the Aspen Center for Physics. I think it was in 1990. And the way I got into computational biology was really a do-it-yourself at-home project. So I realized back in the late 80s that DNA sequences were going into the GenBank at a very rapid rate. They weren't appearing in the protein databases. So really what I decided to do was to load up my first PC at home with GenBank. It came on floppy disks, so I could load it up there. I'd start a 6-frame translation search against GenBank and in the morning, and then when I came back at home at night, I'd take a look at the results, and the results were really interesting and turned out to be actually quite productive endeavor. And then when a paper came out from Ham Smith in 1990, describing a motif finding algorithm, I asked my wife, Jorja, if she could do something with this to make something that we could search to get deeper, to find deeper homologies. And that led to the blocks database, which I presented at that 1990 meeting. And at the barbecue afterwards, after some beers, Temple Smith, who was not really a fan of the Dayhoff matrices at the time, he said, well, you've got your own alignments. Why don't you come up with your own scores? And I thought, oh yeah, that was a good idea. We decided, Jorja got into it and we came up with the BLOSUM matrices, and BLOSUM 62 became the default for BLAST. I think it's still a default. So thanks to Gary and Bill for organizing that and for serving beer at the barbecues. Yeah, but it also told me that, this do-it-yourself-at-home project actually turned out to work out pretty well. It was quite successful. And so what I'm going to tell you about is a do-it-yourself-at-home project along the same lines, but on a different topic here. And the topic is chromatin dynamics. Now, the lab has been studying chromatin dynamics, but what we found is that the kinds of methods that have been available have not really captured the dynamics very well. So from time to time, we've found it useful to come up with some of our own methods, one of which is what we call cut-and-run. We mix live cells or nuclei with lectin-coated magnetic beads. They stick, we permeabilize, we add an antibody, say to a transcription factor. We follow that with a fusion protein to protein, between protein A and micrococcal nuclease or emanase. We then activate the emanase with calcium, and the complex will diffuse into the supernate, and we'd extract DNA, ligate on adapters, amplify libraries for sequencing. And in the variation of this, what we call cut and tag, we substitute TN5 for emanase, where TN5 is the cut and paste transposase. That's the basis for the Illumina and Xterra system and the ATAC-seq method. But in our case, we're tethering such that such that we can activate the TN5 with magnesium, and the TN5 will drop its payload of DNA sequencing adapters. We extract the DNA and amplify the libraries for sequencing. And the backgrounds are very low relative to ChIP-seq, because with ChIP-seq, you cross-link and solubilize the entire cellular contents. But in both of these methods, we only solubilize a little bit of DNA that's in the vicinity of the antibody, and that keeps our backgrounds down. Here's an example of that, where what we're looking at is a histone mark, a mark of H3K27 trimethylation. It's a mark of polycomb silencing. It tends to be in large regions. In this case, in human K562 cells, you see this broad region. Now, because you're sampling from the entire genome when you do ChIP-seq, you're always going to get this low background, and you have to build up the peaks above that. And for that, you're going to need a lot of unique reads, and you're going to need a lot of cells for that. But with cut and tag, the background is really so low that we can go down several orders of magnitude lower and get higher, very high signal noise. And of course, this makes everything cheaper because you don't have to sequence as deeply to get the information that you want. Now, when we were developing cut and tag, we had a problem to deal with. And that is that the protein A part of the fusion protein will do the targeting to the antibody. But the TN5 part is really a problem, an artifact for us, because it'll go to open chromatin regions throughout the genome, and then we won't be able to tell whether it's targeted or open chromatin. But we found that we could suppress that artifact by adding 300 millimolar sodium chloride. What it does is it competes for electrostatic interactions between the TN5 and the DNA, and that suppresses the artifact pretty nicely. So here's an example. What you're looking at is a histone mark, H3K4 dimethylation, that finds enhancers and promoters throughout the genome. And so we line it up over TAC-seq data that gives us open chromatin mapping. And you can see that there's occupancy of this mark, histone mark, about a kilobase or more on either side of the open chromatin mapping, with almost no occupancy over the open chromatin itself. But we realized if we don't add the salt during the tagmentation, what we found was that it's as if all the TN5 got sucked into the hole in between. We call this cleavage under target accessible chromatin, or TAC. And our interpretation is that there's enough flexibility in the chromatin fiber such that if TN5s are attached to nucleosomes in the vicinity of a gap, and if the gap is big enough to fit 2 TN5s and enough DNA in between that we can sequence it, then we'll get a signal, but we won't get a signal in the linker regions nearby. And this predicts that small fragments will dominate, and indeed that's the case. So if we look at subnucleosome-sized fragments, less than about 120 base pairs, we get most of the signal. And we can compare this to TAC-seq for open chromatin by using one of the two replicates from the ENCODE project to call the peaks and the other to line up over the peaks. And when we analyze the data, we basically see that what we're getting is all the open chromatin sites essentially to a high level of statistical significance. So we are mapping open chromatin, but we're doing it using this antibody. And this got us to wonder, well, what about RNA polymerase II, the transcriptional machinery itself? Because we knew from other work that RNA polymerase II will pause at promoters before it moves on. It's a critical checkpoint for transcription in general. And we also know that there's RNA pol II sort of doing the same thing at enhancers as well, not just promoters. So we could we tried RNA polymerase II serine 5 phosphate, where serine 5 phosphate so serine 5 of the heptapeptide repeat present in 52 tandem copies that comprise the C-terminal domain of the largest subunit of RNA polymerase II. Serine 5 phosphate gives us actually the best signal of all. And since we could compare our data to data from ProSeq, where ProSeq maps the RNA base and the active site of RNA polymerase, we could estimate that the distance between the distance between RNA pol2 active site and the midpoint of the fragment is about 130 base pairs or so. What this means is that CUT&RUN gives us accessibility in the wake of pol2, giving us ground truth validation for actually open chromatin. This is basically what we see everywhere where there's open chromatin. And this is going to become important in what I'm going to tell you next. Now, as I was finishing up this project lab, the pandemic hit. The bridge that I take in developed cracks. They shut down the bridge for 2.5 years. And anyway, I was kind of stuck at home. So when the pandemic hit, I grabbed some stuff that, you know, you can buy. This is all surplus stuff, but anyway. So yeah, so this is CUT&RUN at home. And I thought, well, you know, the BLOSUM thing works so well, so why don't we do this? So okay, so all the data I'm going to show you actually was on this setup here. Okay, now what we're going to deal with here is formalin fixed paraffin embedded sections. Now the standard practice, if you go in for a colonoscopy or any kind of surgical procedure, no matter what it is, the standard practice for the surgeon or technician to drop the tissue into formalin, which is at a concentration of 4% formaldehyde. It sits there for a couple days to a week, and then it's embedded in paraffin. It's then placed on a microtome, and slices are cut out and stained so the pathologist can look at it and say, hey, that's what it is. But then the rest of the block is put into storage. And so you can imagine about how many of these paraffin blocks have been accumulating for around the world at all the hospitals and clinics. I would ask around, how many are there? You know, and the kind of estimate I got, like I asked Chatbox, and it said about 500 million a year. And it's been going on for over a century, so never going to change. Okay, so this creates a huge resource for doing things like whole genome DNA sequencing, whole genome RNA sequence. But I realized CUT&RUN actually is really ideal for this, because one of the things that the formaldehyde does is it causes breaks and terrible, you know, so the DNA you're going to get out of small, but we're already dealing with small fragments, less than 120 base pairs. So we're in the right ballpark for this. More importantly, formaldehyde cross-links lysines. Histone lysines are abundant more than any of the non-histone DNA binding proteins. So basically, what the formalin does is to turn your nucleosomes into concrete. But remember I was talking about RNA polymerase II? The C-terminal domain of RNA polymerase II, those 52 copies, do not have a single lysine. So you have the biggest lysine-free desert in the entire proteome. So this gives us something this gives us a way in. We can just use that antibody and carry out CUT&RUN, and that's what I did here. So here we just use break up the FFPE, heat it up, reverse the cross links, and just carry out the CUT&RUN protocol, that CUT&RUN protocol I just described. And the data that we get is really quite nice. So what you're looking at here is the annotations are candidate cis regulatory elements, CCREs. These are put together by the ENCODE project for mouse and human for all the data that they had, mostly from DNase hypersensitivity mapping, which is open chromatin mapping, so it's a perfect annotation for us. And then if we do this on fusion transgene-driven mouse brain tumors here, and there are 2 of them shown here, PDFB and REL-A, and look at RNA pol2, you get these really sharp peaks. We can also do this with H3K27 acetyl. It's a histone mark. So whether the epitope is inside or it's on the outside coming in, we get exactly the same very sharp peaks. And we compare it to other methods. FACT-seq is also based on CUT&RUN from a different group, and they also did CUT&RUN frozen or compared ChIP-seq. And the signal noise is really through the roof. Okay. So now what we've done is to make the method even simpler, so a high school student could do this. And basically what we do is take the slide, stick it into crosslink reversal buffer, heat it up to reverse the crosslink. The paraffin floats off, and now we just drain it, put a little antibody on, cover it with plastic wrap, give it a wash, do the next antibody, et cetera, up to the point of tagmentation. So after tagmentation, we can scrape it into a tube. We can also dissect it. Here's an example from a REL-A mouse, a transgene-driven mouse glioma. What you're looking at are different sections, 10-micron sections, and it's stained, H&E stained, and this is the kind of thing that pathologists look at. So you can see that, wow, this tumor is really loaded with nuclei. Normal brain is really kind of distant. Okay, so it was simply a matter of just scraping each of these sections, as indicated here, tumor T for tumor, N for normal, into 6 different tubes. And that one slide generated this volcano plot where the top 3 based on fold change going up is the collagen gene. And if we look at the tracks, you can see that RNA Pol2 is really loaded over the collagen gene for the 2 tumor sections and not at all for the normal section. These are just replicates. On the other side of the ledger, we have 2 genes, and they're on different chromosomes, but they have the same name, MER124A. And if we look at the tracks, we see over the MER124A 1HG gene, we can see the inverse pattern. MER124A is a microRNA, 22 nucleotides, and it is very well studied, the most well-studied microRNA in the brain, in the sense that it controls development in invertebrates. It's highly conserved. It's actually in glia in C. elegans, the worm, and also controls development. The same 22 nucleotides controls neuroblast proliferation in Drosophila, and it's also a cancer risk marker, and also it's a known tumor suppressor gene in metastatic melanoma. And with H. pylori infection, it actually is a DNA methylation marker, is already known to be that. We're looking at the other side of the ledger. This is what we're looking at, RNA Pol2. This is what DNA methylation is actually shutting down. However, nobody knew about this because it's undetectable by RNA-seq, and everyone's doing RNA-seq, and nobody realized it. Because for RNA-seq, you need the prime, and this is very small, and there's nothing really to prime on, et cetera. Okay, that's one thing it could do. But something else it could do that I think is much more interesting is that we can use this to detect hypertranscription. Now, hypertranscription, by definition, is proportional upregulation of transcription. And from the title of this paper, Widespread Hypertranscription in Aggressive Cancers, this is something that maybe cancer clinicians should know something about. Because in this paper, they were able to measure hypertranscription and showed that it confers a worse prognosis, regardless of all these other factors, just everybody going up. And this is based on a large cohort. The way, I'm not going to go into how they actually did this, but it was very difficult to do this because when you're doing RNA, they did it with RNA-seq. If you want to look at hypertranscription, you like to look at nascent transcripts, but they're looking at stable transcripts, and each of the transcripts have different half-lives and things like that. They needed really a large, in order to make this claim, they really need a large number of, they need lots of samples, which they had from TCGA data. Okay, so, but I realized that actually we don't have to go through all that stuff. For one thing, we're right on the DNA, RNA Pol2. So think of it this way. In a window, if you're looking at one, where there's one RNA polymerase 2 in any particular window of time on the DNA, if you hypertranscribe, say 2-fold, there'll be 2. If they're 2, there'll be 4. They're 4, there'll be 8. Basically, that's what hypertranscription is everywhere. Okay, and we can do that in a very simple way. And actually, this is where Jorja came in again. She just discovered something been around for a long time, something called Bland-Altman plot. Let me just explain what it is. If you know addition and subtraction, you can do this. Okay, so we take each of the CCREs, and over the span of the CCREs, we're going to add up the normalized counts. Doesn't matter how you normalize it, just so you do the same for the tumor and normal. We're just going to add them all up. And then we're going to subtract the normal from the tumor, and we're going to plot it as a function of the ratio, the function of the average t plus n over 2. And just for clarity, I'm going to show this on a log scale. And what we see for rel A, the vast majority of the RNA pol2 is in the that's engaged in the cell is hypertranscribed because it goes up. It could have gone down. And in fact, if we look at the others, well, here's from a mouse brain. This is one mouse where it shows strong hypertranscription. Here's another mouse that's very weak. So very personal hypertranscription here. And for YAP1, we don't see any at all. Okay, this is artificial cancer. Let's look at some real cancers. Now you might think, so I basically bought some FFPEs from a company, and they sent something. I thought, oh God, this is really small, only 5 microns. I wanted 10, and they're only about they're really small. Nevertheless, we have tumor and normal, and just think of it as a random sample of cancers throughout the human population. And lo and behold, what we see is that 5 out of 7 of this random sample show clear hypertranscription. It's only the kidney and the tumor that we don't see the hypertranscription on. Now, what we next noticed was if we just take the top 20 of the million CCREs throughout the human genome, we take the top 20 and just ask where they are. 18 out of 20 are in chromosome 17. What is going on here? Not only that, most of them are either at 33 million on the map of chromosome 17 or 37 million on the map. So I just write down the names type the name into PubMed and the word cancer, and lo and behold, ERBB2 really stands out. It's 35,000 publications on ERBB2, and that's about 2 orders of magnitude more than the next highest one is CDK12. Okay, well, ERBB2 is, of course, well studied because it's responsible and the targeted therapy for about 30% of breast cancer patients. And they mentioned here amplification. Thought, oh, amplification, let's take a look. And lo and behold, in 2 of the patients, one breast, the one breast and one colon, you can really see that there's this overall region that is broad, that's covered compared to, okay, so we're seeing amplification here. It's pretty clear amplification, but we also see that there are these sharp peaks. So what are those sharp peaks? They would be promoter peaks, but they're embedded in this large region. And so what we did was we just tiled, we just made bins every 1 kilobase over a megabase region and did some curve smoothing. And what you see here is around ERBB2 or HER2. And you can see the peak at ERBB2, but we also see some satellite peaks. And let's take a look at what they are. So the satellite peak on the left is the most interesting to me. And that is bidirectional promoters for MED1, which is mediator, the largest mediator subunit, and CDK12, which I'll get to in a moment. And you see that only in the breast. We don't see it in the colon patients. So very personal. Now, if we look more closely, what you see, particularly for ERBB2, is you have these peaks throughout, and they're all sort of adding up to give you this broad region. And what this indicates is something that's actually been known, as this doesn't show up. This is a 1976 paper showing that clonal selection in tumors. It's actually very well studied. Okay, this shows up here. This is a recent review showing that extrachromosomal DNA in cancer occurs, and this has been very well known for decades, via these double minute chromosomes which partition unequally at every cell division. And so that can give you these selective sweeps. And so what we're basically inferring from our data from these 2 patients basically is amplification followed by selective sweeps. Now, in particular, CDK12 and MED1 have been shown to independently, so in the context of a HER2 amplification, when they are amplified as well, that predicts poor prognosis in both cases. And so we wondered whether if we could actually test this idea, and I'm going to get back to basically the transcription cycle. So let me just explain something. In order for pause release to occur, so the pause PAL2 I showed you has serine 5 on it. In order for it to occur, the RNA polymerase machinery has to release from the mediator complex. MED1 is the largest subunit there. But more interestingly, the serine 2 phosphorylation has to occur on the CTD. And that's mediated by a few different cyclin-dependent kinases. And the one we want to think about here is cyclin 12. As its name implies, the kinase has a cyclin partner in cyclin K. So it's a matter of seeing whether this makes sense that cyclin CDK might be sort of giving push to the polymerase to give you hypertranscription throughout the genome. And indeed, what we see is when we look at cyclin K, it's upregulated independently. It's on a different chromosome that we see in the breast, but not in the tumor. So let me just summarize what I just described. I showed you that we can do profiling with CUT&RUN, cut-and-tag, CATAC, that pause-PAL2 maps the transcriptional regulome, that FFP CATAC identifies tumor suppressor microRNAs and reveals hypertranscription in cancer, that our model is CDK upregulation drives hypertranscription. And we can test this just by doing the same thing on the tumor repositories. That would be the best way to test it. And we're doing that right now. So I'll stop there and acknowledge people in my lab who've been involved in the project, particularly over on the right. You see Jorja, Yizhang next to her, and Kami Ahmed. And I'll stop there and take questions. Thank you.
Hi, that was interesting. How do you distinguish hypertranscription of a single locus versus like?
No, hypertranscription by definition is proportional upregulation at all those sites. Okay. What you just described is upregulation, and that's not what I'm talking about.
Thank you.
Got it? Okay.
Thank you. My question is, how good is it to interpret like the repetitive sequence binding affinity? Do you have any plan to come up with new protocol so we can look at repetitive sequence?
You might, so if you want to try it, here's a QR code that goes to the protocol. It's really easy to do. Like I say, I do it at home. We're actually preparing to teach it to high school students because FFPEs are really cheap. You can get them and they can learn real biology by learning about the transcriptional apparatus. So, I think I don't see any improvement of it. I think it's ready to go for, I should mention, we are scaling it up and we've automated cut and tag in our shared resource, and there are companies that also have it automated. And this should fit in. We have it working on curls, which are really fairly inexpensive to get. And it's really ready to go for high throughput and can be done not just on on archives, but it can be done, I believe, in rapid enough time that it could be used for clinical diagnosis.
Steve, so to achieve hypertranscription, the numbers I carry around in my head is that there's not like a huge pool of available RNA, like free RNA polymerase. So do these cells just like fully engage the free ones, or are they also making a new big pool of PAL2?
Okay. I don't have time to get into this, but let me just show you something. Okay. We focus on the histone locus body. This is the only thing that matters, okay, for hypertranscription. The histone locus body is not familiar to people, but in fact, all of the replication coupled histone genes are here. Now, MXC in Drosophila, this is from Drosophila, MXC in Drosophila is the structural component that holds the whole thing together. So it's a nuclear body, it's got all the histone genes that are made of replication, specific for S phase, only at S phase, it actually gets bigger. Now, this didn't show up, but anyway, So in Drosophila nerve cells, you get these rapid divisions. And if you look over the histone locus body, you see that for PAL2, you see that it's as if most of the PAL2 is actually over the histone. I mean, there's a huge amount, but there's only 1/100,000th of the entire genome. And so let's go through the numbers here. So proteins, proteins about 40 to 50%, RNA about 4%, DNA is 2%. Histones need to make 4% of the total protein in S phase. So if you go through the numbers, what you realize is only a third of the cell cycle. There are 10,000 protein coding genes that are active. You only have about 100 histone genes to deal with. So you have to put all your PAL2 over this. And that's what gives you what gives a cancer cell the advantage. You need 100 times as much engaged RNA PAL2 for histone genes to make 4% of the cell's total protein a third of the time in order to wrap all the DNA, otherwise you'd be dead. So I think we have a good model for hypertranscription. What's causing it, I think it's just the histone genes, because otherwise, you're already high, make a little bit more, how that's going to help. It's really going to help for the histone genes.
It's a very nice talk. I have questions about the therapeutics. If we want to target the hypertranscription, should we target the histones or the specific protein like the regulators like CDK9, or should we target the transcription components like the DNA transcription or DNA replication in this machinery? How we want to target the hypertranscription? What is the better way? Thank you.
Well, one way, so one thing, we have shown that, so doxorubicin is one of the most widely used drugs for given to cancer patients, but and there's others, and that can cause DNA damage because it poisons topoisomerase II, cause DNA breaks. However, we and Jacques Niftis and others have found that actually the anti-cancer effect is mainly through disrupting chromatins, basically disrupting nucleosomes. And so there'll be a trends in cancer review from Nefjust, Katja Garova, and me, where we basically point out that if you think don't think about mutations, think about the disruption of chromatin that occurs that can drive cancer. And these are common chemotherapeutic drugs. So improving those drugs is really, we think, the way to go, as opposed to some kind of targeted therapy where, a single mutation and now basically it's gone. They're all done using normal cancer chemotherapy. So I think the focus really should be on improving the general cancer chemotherapy drugs to get at this problem here.
Thank you very much.